Hi, this is Christoph, and I will give you a quick walkthrough through the microRNA sequencing data that you received from us. You will find them either on a DVD, or if the experiment had a larger number of samples, they will be on a flash drive that are included with the report. And of course, you have always the option to download them from our FTP server. So here are the files shown and the folders shown on the FTP server. You have the individual sample files. They each contain the raw a subfolder for the raw data and for the process data. Then, so you have the 6 individual sample files here in this case. Then the important folder that includes all the analysis is the comparison folder. So this folder typically is compressed on the server, so it's easier for you to download. So you'll find it only as a zip file. And this one doesn't contain the raw data any longer, but the combined process data. The README file is just a file briefly explaining which folder includes what, and the appendix includes additional technical details. I'll go through that in a little bit. So I downloaded the comparison folder already, and I will go through the individual files that are included in this folder. So the README folder, like I said, includes just the brief description of each folder content. So then the comparison folder has subfolders, of course, the process data, map data, then the data summary. And if you had biological repeats, then there's also an in-depth data analysis folder. So let's first go through the brief report, to the overall experimental report. Just to mention, all the data that I'm showing here, all the files, due to size, to keep it simple, we truncated the data. So where you see here sometimes in the Excel spreadsheets only 5 lines, you would have thousands of lines. So let's take a look at the demo report. So on the first page, it basically mentions the number of the different samples, species, which database was used for the mapping. Then the terminologies used, explanation just of some of the terms. Then just very brief method and procedures. And then we come to the actual results. So in these pie charts, what is shown is first the number of raw reads, and then the different filters that were applied. So first we check whether the adapter was found or not. If not, then they will kicked out. Then the second filter is basically a size filter. If the reads are too short, the sequence we found too short, then those will exclude it. And then the so-called junk reads are basically low complexity reads that will be excluded in this step. Then the second pie chart shows you the number of mappable reads, and you see here in the legend the color coding for different groups. And what these different groups are is shown here in the next flowchart on the next page. Basically, when we map the reads, the first thing we do is we try to map the reads to miRBase. So this is basically the top half of this diagram. So if we're able to match them to any sequence in miRBase, that's great. So this will be group 1 through 3. If there's no match in miRBase, we see if it can be mapped to the species genome, and if so, they end up in group 4. And then there's an important distinguishing factor here between group 4A and group 4B. Group 4A is the very interesting group, which are basically potential microRNAs, because the extended sequences can form or are predicted to form hairprints. Group 4B, on the other hand, can be excluded as potential microRNAs because they cannot form hairprints. Then here's just an overview again over the files that are included in the folders. Then the next chart shows you the length distribution of the mappable reads. The same information just presented as a table. And here the quality scores, the Phred scores for the reads. And here in the last page is in table 6 are the basic statistics shown for the different groups, how many reads, how many mappable reads were found, etc. How many in table 7 then goes into details about the known and predicted microRNAs and which group exactly had how many unique microRNAs. Okay, then that's the demo report. Then let's look briefly into the Excel spreadsheet that comes with it. So on the first tab here, the read summary, it shows you the raw reads, number of mappable reads, etc., which ones were excluded for the different samples here. On the next tab, what is shown here are basically the raw and normalized data number of reads for each microRNA sequence that was found. So here in column B, you see the microRNA sequence, in C, the microRNA name, then to which group this was classified. So in this case, it was, of course, we could map it to miRBase, so it's in one group, in group number 1. And then you see the raw reads for the individual samples as well as the normalized reads. So this is the raw read for sample 1.1, and here these are the normalized data for 1.1. What can get confusing is we separate the results here based on the abundance. So the if you scroll down here, the number of reads of the following report microRNA is less than 10. So 10 is basically the detection limit, the cutoff that we set. So if we get less than 10 reads for a particular sequence, we list it down here. And then the ones here at the very top show microRNAs that have reads that are above the average of the data set. And then here below this line, those that are below the average number of reads of the data set. Okay, then if you go through here, basically just briefly mentioning the folders, process data are in here. Map data for the individual groups are listed in folder number 3, the map data. And there you have basically for each group you have two files. One shows you the alignment. So basically you have the genome listed here, and then it shows you the alignment of the microRNAs and the reads that were found. So this is just separated for each individual group. And then you also have a summary file for each group, which basically gives you more in-depth statistics about the number of reads, how many microRNAs were detected, etc. And then in folder number 4, it contains the summary file, of course, which includes the unique microRNAs of all groups, including group 4A that I mentioned, that includes the potential microRNAs. The only data that are not included there is group 4B, which can be excluded as a potential microRNA. Then if you had biological repeats in your experiment, you will see a folder called in-depth data analysis. And if you look at the at this report, it basically depending on the number of samples and of course the instructions that you give us, how you want the comparison to be done, you will find ANOVA and or t-tests. So the format is always the same. Initially it shows you which test was run, how the samples were grouped. In this case we had 2 repeats, 3 groups, and then on the next page the heat map is shown with different p-value cutoffs, and you can actually click on these and get a better view. Then on the next page you will find, and again this is truncated, that's why you see the periods here, a table with a microRNA name, the p-value, and the number of reads for each individual group. And on the next page you will see a heat map for all detected microRNAs, independent from the t-test or ANOVA result. So here the same thing with the t-test, where you have only 2 groups, heat map, and the table that shows you microRNA name, p-value, read, mean read, number of reads, as well as the log2 ratio, which is basically indicates the fold change. And just to let you know, you will find the high resolution individual heat maps in the individual test folder, as well as the full t-test results. That is also shown in the PDF file. Okay, this is pretty much it. I just want to highlight a couple of questions, frequently asked questions that we receive. One is regarding the normalization, how we normalize the data. For that, I would have to refer to the appendix. On page 6, it is here under point 4, multiple sample comparison. You will find the normalization method and the formula used. So this is in there. Then another question that we frequently get is in the in-depth data report. When you look at the heat maps, you see up here this color key, and oftentimes people are confused what this stands for. So this is not representing any reads or so, but the so-called Z values. And again, I have to refer to the appendix. The formula for the Z values is mentioned in there. Then when you look at the demo report here, or the actual data that you get, and look at the microRNA names, you see sometimes microRNA names that are not specific for your species. And so that confuses a lot of customers as well. So this happens if you basically during the initial step, when we try to map the data, we will map them to miRBase. And if there's a microRNA that is not discovered and not described for your species yet, however, it is, let's say, described for human, and you work with mouse, and has not been described yet for mouse, then it will be shown here as the human, with a human name. And in general, when you go through the reads, you will also sometimes find reads for example, for messenger RNA, and this can happen simply because we do a size exclusion, of course, but if you have degraded messenger RNA, it can of course end up in your library, and that's why we filter for it. So I think that's should cover most of it. If you have any questions, give us a call or email us at info@lcsciences.com. Thank you.